Welcome back to the channel. This video is episode 4 of my May 2021 IB Diploma Mainstream Science Multiple Choice series. And here I'm recording myself doing the Time Zone 2 Biology Multiple Choice paper. So far, I've done both chemistry papers. I have done the Time Zone 1 Biology paper. My average so far is 39 out of 40, which is very good if you are a student. But then I am a teacher/tutor. And I have a PhD in science doing high school kid things. And I have a lot of pride, like showing off my STEM knowledge. So 39 out of 40 is definitely a score, an average score that I look forward to improving upon. The real purpose of this video, however, is for me to demonstrate my problem-solving skills and thought processes to high school students who otherwise feel that science is difficult for them. So let's jump into the paper. So this is the paper. I need to get my snipping tool ready as well. And this is my Excel, which I'm going to record my answer on. Already question 1, I'll make it slightly bigger. The image shows an electron micrograph of some virus particles or some bacteriophage, if you recognize the shape. What does a virus have in common with a living cell? Viruses don't have ribosomes. Viruses do have genetic material, so most likely B is correct. Viruses don't reproduce by binary fission. Viruses reproduce only using host cell like material synthesis mechanisms, and viruses do not respire. So answer should be B. Second question. These 3 cell types are shown in the micrographs, and you're supposed to distinguish between them, I guess. If you have revised enough, then I think you can recognize that cell A is like mostly a white blood cell, a B cell, B lymphocyte with lots of mitochondria and a big nucleus. The second one should be I can make it slightly bigger, I guess. Yeah, B is definitely a photosynthesizing cell, and then C would be a I was about to say ciliated cell, but this looks more like a cell that lines the villus. The structure on the outside looks. I'm not. I think actually I may feel that that's actually more like a ciliated cell, but because the villi brush border does not look like this. Most likely you don't need to worry about that particular detail for now. Which feature distinguishes the striated muscle fibers from the 3 cell types shown above? Well, striated muscle fibers would represent an exception to the cell theory with multiple nuclei. So you just need to recognize the correct answer. A popular exam multiple choice strategy for a student would be to rule out the wrong answers. But that's kind of time consuming. And that's usually something that like physics student will try to do. But I think that's a poor strategy. You may be able to get a 6, but you won't be able to get a 7 because you'll end up wasting a lot of time ruling out the wrong answers. So if we just solve the problem and recognize the correct answer, just choose it and move on. Question 3, which graph best represents the relationship between concentration of chloride ions in the external environment of a cell and the rate at which chloride ions move by facilitated diffusion into the cytoplasm of the cell? So the higher the external concentration, by higher external concentration represents steeper concentration gradient. So the rate should definitely increase. Facilitated diffusion, however, is a process that requires protein channels. And if the channels are all being in use at a particular moment, then you can have even higher concentrations, or even steeper concentration gradient. But then the limiting factor would be the availability of chloride channels. So the answer should be C. Question 4, when does DNA replication occur? So G1S, G2 would be interphase, and I think this is just a matter of knowing your stuff. So G actually does not stand for growth, but that doesn't really matter. It's the gap phase, I think, where a cell actually grows and do its own thing to serve the organism. But S standing for synthesis is the time when DNA replication occurs. 5, which process is an example of catabolism? So catabolism involves the breaking down of complex molecules, often by hydrolysis. And guess what? C is the answer. And all 3 other choices will be examples of anabolism, which involves condensation reactions. 6, the diagram shows a part of the molecule, and this looks like a branched polysaccharide. Amylose is a polysaccharide, cellulose is a polysaccharide, but they are unbranched. Collagen is a protein. Amylopectin is your branched polysaccharide. So answer is D. Question 7, which organic molecules may contain the element sulfur? Has to be proteins, because proteins use amino acids, and 2 of the 20 amino acids contain the sulfur atom. Carbs, phospholipids, and nucleic acids don't contain sulfur. Moving on, question 8 of mesosalt and stall experiment. Hopefully students would just recognize the correct, or recognize the entire, remember the entire process, and the experimental result proves that DNA replication is semi-conservative. I hesitated for a moment because I thought 3, 4, 5, and 6 represented different generations, but turns out those are just the choices that the student we have to choose from. But let's see what they're asking. So which centrifuge tube shows the arrangement of bands observed after 1 round of replication? It should be 6, because all the helices will contain 1 heavy strand and 1 light strand. Let me read the question more carefully. N15 transfer DNA bacteria produced as a result of replication. Yeah, I think this is just standard treatment. So 6 is definitely the correct answer, because the N15 parent strands were used to 6. No, actually, I mean D. So each N15 parent strand used as a template, and each helix contains 1 entirely old strand and 1 entirely new strand. All right, sorry for wasting time here. Question 9, what does the RF value in thin layer chromatography represents? So chemistry student probably knows it very well. It has to be a ratio of distance traveled by the compound of interest versus the distance traveled by the solvent, which otherwise carries, or which is responsible for carrying the spots up the chromatography paper against its gravity. Question 10, which is a feature of the human genome? I want to rule out the RNAs, because RNAs are not permanent genetic material. Mitochondrial DNA should be part of it. This is a weird question, but I think I will go for mitochondrial DNA. Because you make mRNA based on DNA as a template, but and mRNA is not permanent, and transfer RNA, that's not really code for any proteins. They help the synthesis of proteins. But messenger RNA do help the synthesis of proteins, but I would just go for mitochondrial DNA for now, and hopefully I don't embarrass myself. Question 11, what feature of eukaryotic chromosomes distinguishes them from the chromosomes of the prokaryotes? Eukaryotic chromosomes definitely have histone proteins. They're not circular, they're more like linear. Double-stranded is true for all kinds of chromosomes. Multiple genes along the length of each chromosome is not true, because even the circular chromosome that bacteria cells have contain multiple genes. Question 12, a tetrad is a group of 4 cells produced when 1 mother cell divides by meiosis. The tetrad indicated by the row contains a total of 48 chromosomes. So each cell contains 12. And by the end of meiosis, the cell would have divided twice. So 12 would be the haploid number, which means that the diploid number is just 24. Woman with blood type A has 3 children with a man who has blood type AB. The first child has blood type B. Well, for these blood type things, the probability of the first child has no influence on, or the blood type of the first child has no influence on the probability of the second child, except that I think you need to infer that this missing allele from the woman has to be an I allele. Because blood type A, well, being blood type A means that you can be homozygous for the A allele, or you can be AI. And this woman has to be AI because there's one kid who has, well, the first child has blood type B, Like, had the mother been homozygous A, did I say heterozygous at first? But I mean homozygous A. Had the mother been homozygous A, there is no way that the first child would have blood type B, which means that you have to, that means that you can infer that the mother must be heterozygous. And now if you complete the Punnett square, the chance that the second child has a blood type AB would still be, well, 1 in 4 instead of the 1 in 2 had the mother being homozygous. Question 14, which organism would be classified as a saprotroph? And saprotroph is just a fancy term for decomposers. And the typical decomposers are fungi and bacteria. So C looks promising, but let me read the rest. Single-cell eukaryotes are rarely decomposers. Yeah, photosynthesis, that's an autotroph and a producer. Ingestion, no, definitely not. That's a heterotroph. CMOS is correct. Feed by secretion of digestive enzymes on its food and absorption of the digested material. Yes, that's exactly what a decomposer does. And choice D, by the way, would be the detritivores. So they do have a digestive system. They do eat. Question 15, the diagram shows the flow of energy. And it's a big diagram, so I need to make it smaller. Oh, that's so big. Big diagram. So what percentage of the energy passed from the primary producers to primary consumers is lost to cellular respiration by the tertiary consumers? That's crazy. So much information. On the left-hand side, the tertiary consumers loses 80 units of energy, while the primary producer passes on how much? Well, passes on a lot here. 40,000 units. So I'm guessing you had to do 80 divided by 40,000, and I think that's 0.2%. Weird question where they just gives you a whole bunch of numbers. And the 10% rule does not really apply here anymore when they give you exact figures. And I hope that's what they want. Now question 16, what gases have made the most significant contribution to global warming? Carbon dioxide is the most famous one, but then the second gas, water vapor is a very significant greenhouse gas. But that's a natural greenhouse gas. Is that what they want? Because methane is another significant greenhouse gas. So contribution to global warming. I think global warming is defined to be the result of the enhanced greenhouse effect. So water, which is mostly responsible for the natural greenhouse effect, is probably not correct. I'm not sure about this one. Let me just color code the ones that I'm not sure about. And then 16, I'll go for B for now. Ozone is not famous for its greenhouse gas effect. 17, which evolutionary pathway is most likely to result in the evolution of analogous structures in species W and Z? So they have different ancestral origins, but then because of similar living conditions, they develop structures that have similar functions. That's weird. I think C looks the most reasonable because they don't belong to the same lineage. B would be the answer had they been asking for homologous structure. And yeah, A and D just both look weird because they belong to the same lineage. So one species evolves from the other one, but that's not what organisms that have that share analogous structures are supposed to evolve. I'll just go for C. 18, this is a pine cone. So it's a conifer. So just A. 19, what information can be deduced from the sequence of nodes in a cladogram? So each node is represents an event where some basically represents a divergent event. In a cladogram, based on what information? with the sequence of nodes, okay, not the number of nodes. So the number of nodes may be more related to the number of mutations. Not the period, because it has to be the position of the nodes. Like if they drawn the cladogram, like the branches to scale, then yes, then period. But I feel like B is the most probable answer. Yeah, the sequence of divergent events. I'll just go for B. It's a weird question. So this is like evolution is literally like the most like annoying topic because the answers are so like vague and ambiguous. Okay, 20 in chemistry is super easy. This is hydrolysis reaction converting things into, well, converting a polysaccharide, 4-unit sugar into 2-unit sugar. So that's definitely amylase, digesting polysaccharides into maltose. So these two sugars represent maltose, which are themselves digested by maltase. Glucosidase. I don't think this is actually an IB paper. Is it an IB paper? All enzymes are, well, all like sugar hydrolyzing enzymes are glucosidases, I'm guessing. But A is definitely more correct. So I'll just go for A. 21 cardiac cycle, they're asking for, at what point during the cycle does the atrioventricular valve close? So when the AV valve closes, well, the AV valve closes because the ventricular pressure builds up. Well, because it's time for the ventricles to contract. And when the ventricles contract, it will first push close the AV valve and then it will push open the sublunar valves. And yeah, when the ventricles naturally contract such that its pressure exceeds the atrial pressure, that's the time when the AV valves close. So A should be the answer. 22, what's the role of antibiotics produced by fungi? I think B should be the answer. Antibiotics target bacterial metabolic pathways, bacteria, prokaryotes. 23, where in the body are type 1 pneumocytes? And you just need to recognize that type 1 pneumocytes are the typical epithelial cells which facilitate lung gas exchange. So where's A? Electron biograph shows a transverse section through a myelinated neuron. What process is facilitated by the presence of structure labeled X? The myelin sheath will allow the signal to, will allow the flow of ion to remain so far intense. Being more insulated by the aqueous surroundings, most likely will reduce the attractive forces exerted by the surrounding water molecules on high concentration of sodium and sodium ions, which themselves are polar. But that's how insulation works, I guess. I'm not sure whether that's a legit explanation, but that's what I tell my students all the time anyways. But either way, the insulation allows saltatory conduction, which otherwise occurs at the nodes of Renvia. Well, saltatory conduction should be like the entire event where you have super fast nerve impulse conduction in the myelinated sections like this. And then at the nodes of Renvia, that's where all the sodium potassium pumps and all the voltage-gated ion channels do the thing in allowing more sodium to enter. Anyways, 25. What's the similarity between the testes of males and the ovaries of females and humans? They produce gametes throughout the life. That's not true for the female structure. They secrete hormones into the bloodstream. That seems reasonable because testes release testosterone, and then the female ovaries will release either estrogen or progesterone at different times of the menstrual cycle. Let's look at C and D first. The development is controlled by the gene on the Y chromosome. That's not true for the ovaries. They release products to the outside of the body directly through the urethrum. That's true for the guys. The urethra is the common duct for urine and semen. Females, no, I don't think so. So B is definitely the correct answer. Wait, I think I need to. Do I need to color 20? No, you know what? Let's just move on. 26, okay, Hershey and Chase experiment. Percentage of isotope remaining outside the cell. So that was the experiment that shows that genetic material is DNA, which is phosphorus rich, is the genetic material. And a lot of the genetic material is injected into the interior of a whole bunch of bacteria that were invaded by some bacteriophage virus. And the bacterial material shown inside the pellet, or like the sediment. So H, the percent of isotope remaining. Oh, that's interesting. There was a lot of phosphorus in the pellet, so I'm guessing X is the pellet, which they call the sediment. Sulfur is mostly in the supernatant. So well, B and C are clearly wrong. Well, no, A may be correct, and then B may be correct. C and D are clear. No, wait. No, B and C are clearly wrong. Well, D is clearly wrong, so that can't be double sediment. Okay, so it has to be. Well, can it be double sediment? Well, actually, read this again. So percentage of isotopes remaining outside of cells. Well, samples. Okay, let's read the question. This is annoying. So samples collected from the suspension were centrifuged to record the percentage of isotope remaining on the outside of the cells. Most of the sulfur remains outside. Suspension of the infected agitated with the blender and samples collected from the suspension centrifuge, because there was a liquid. Wording's so confusing. When they say outside the cell, do they mean is it like attached to the exterior of the cell, or is it actually the supernatant? That's something that bothers me right now, and it's most likely something that needs to be figured out as well. And the supernatant is a liquid for sure, and the sediment is going to be the pellet. But then when the material is outside the cell, it's still attached to the cell membrane, more like the cell wall, which is the exterior of the sediment. Well, there was no X outside of cells, and by the end, there's a lot of X outside of the cells. But then they're still asking for phosphorus in the sediment. Percentage of isotope remaining outside the cell. Sorry. Why would it start with 0? Because it's the viruses that were radioactive and they were outside the cells. And the viruses were radioactive. Okay, I'm guessing the suspension was already. So they try to analyze the already infected bacteria. I hate it, just spending so much time. So bacterial cell. So we know that the radioactive sulfur is here and the radioactive phosphorus is in here. You blend, you basically shake off most of the sulfur. So I'm guessing X can represent the amount of sulfur in the supernatant. And then Y can represent, well, if it's increases, there has to be in analyzing the supernatant. Yeah, because the centrifuging process and the shaking process will cause some of the protein coats to be shaken off. So most of the sulfur is shaken off. And can there be a lot of the phosphoruses in the supernatant as well? It's so confusing. Okay, I'm just going to go for B now, contrary to what I said at the beginning of the video. So it was confusing. Okay. We may even come back to check this because I really want to do well in this one. I need to improve on my average. Okay, 27. What's the feature of transcription in the single cell organism shown in here? Single cell organism shown in this micro. This looks weird. 1 μm. This looks like the complete cell. That looks at the nucleus. It's definitely not a bacteria. So transcription. This is so weird. So that's called an equal binding, except not true. That's translation. If I zoom closer into the thing, does it actually make a difference? Because I don't think the micrograph shows anything important. Because mRNA splice, so this is the nucleus. But with, you can clearly see like membranes inside those. That's not a bacterium. It still looks very weird. You remove introns to make exons, but the mRNA splicing, I can't imagine this being a bacteria. Could this be like a mesosome? Is that just because of the folded in membrane? The mesosome is not really in the syllabus. But because if A is correct, then B is for sure correct. Maybe this is just a bacterium. Okay, then this structure is a mesosome. And yeah, the nucleus looks more grainy than this. And if you go back to question 2, you can see the structure of a nucleus of cells. They're more like grains and dark and light spots in there. So that structure enclosed in this blob here is most likely not the real nucleus. So D would be true had this been a bacteria. So hopefully this is correct, but for now, I think I have to orange this. Okay, 28. mRNA codon UAC codes for the amino acid tyrosine, which tRNA carries tyrosine. Oh, that's interesting. So if we do complementary base pairing first, well, actually we can already rule out A and D because there's no way that there's a T on the tRNA. And if the codon UAC, then the anticodon would be AUG. And I thought they're going to test you on the directionality, but clearly, because the tricky answer would be, the tricky choice would be GUA instead, but here clearly B is the correct one. Okay, C, curve showing the effects of a non-competitive inhibitor. So a non-competitive inhibitor will cause the enzyme activity to decrease because a non-competitive inhibitor just binds to the enzyme at the location other than the active site, but then the binding causes some deformation of the active site shape that resulted in the enzyme less active over all concentration. So the answer should be B. A would be the other legit graph, but then that would be the competitive inhibitor, and C and D I've never seen before. 30, what molecule functions at the final electron accepted in the mitochondrial electron transport chain? And this you just need to know is oxygen. 31, the Hill reaction occurs when isolated chloroplasts are exposed to sunlight in the presence of DC-PIP. And DC-PIP replaces NADP as the final electron receptor. for light-dependent reaction. So you just need to know the general function for NADP. So NADP accepts electrons from the water itself, becoming NADPH. But if we replace NADP, then that means that the light-dependent reaction can still take place. Then most likely it's just going to be oxygen and ATP. So basically the same answer. It's just they're just a really convoluted way of asking what's the product of the light-dependent reaction in general, because the role of DCP and NADP are similar. Okay, 32, transpiration, I'm guessing. What factor would be a controlled variable in the experiment designed to assess the effect of temperature on transpiration rate? Opening and closing, you can't really control that manually. The light intensity should be controlled for sure, but let's check the rest. The height of the water is not going to affect the rate of transpiration too much. I guess just most likely you're going to look at the rate, average some marker, such as like a bubble in a stream of water, or maybe the calibrated pipette. Actually, the calibrated pipette would it have some physical effects? Probably not as much as the intensity of the light. Evaporation of water is definitely not a controlled variable. It's something that will happen. In fact, it's more like the extent of evaporation is reflected by the drop in the water level in the calibrated pipette. So D is more like a dependent variable. 33 is a section through the stem of a non-woody plant. Interesting drawing. Where does transport of sucrose occur? And you just need to recognize where is the phloem. The big opening represents xylem, and then the top is most likely the bark region. And the phloem should be D, because this entire structure will be a vascular bundle with the phloem closer to the exterior. 34, how does oxygen contribute to phototropism? It increases, well, more like they're transported by, well, their transport is like the result of light-sensitive proteins, which are the phototropins detecting light, mostly blue light, but I don't think A is correct, because oxygen in the presence of light-sensitive proteins get transported to the dark side. So I think B is reasonable. If they change this to elongation, I think that would be better. So I just go for B, and because C and D don't look too legit anyways. 35, okay, topic 10. What process occurs in both mitosis and meiosis? Chiasmata is only for meiosis, reduction division only for meiosis. Chromatids do separate in both, in mitosis and also meiosis too. D is only for meiosis. And I think this question also shows in paper 1, it's phrased in a different way. Wait, I typed wrong, okay. Okay, 36, which cell is a polyploid zygote produced by fusion of one haploid and one diploid? So you should have 3 pairs. 3 copies of each chromosome. So you're looking for multiples, 3 for sure. A and D both have 3 chromosomes, but then A, you need each chromosome to appear, each sized chromosome to appear 3 times, because typically the 2 chromosomes with the 2 homologous chromosomes, but here this is a triplet. 37, this graph shows the changes in concentration of an antibody in the blood of an individual subjected to 2 separate infections. What distinguishes the event of occurring at Y from those occurring at X? And X looks like it's the first time that the individual is exposed to the pathogen. So that's a primary, this bump is the primary response, and slower and also less, results in lower concentration than the secondary response. B looks highly legitimate. If the first infection, there's no memory cells yet. Future infection, the memory cells are there. And that's also the principle of vaccination, like COVID vaccinations these days. Yeah, the rest is just wrong. 38 is to recognize the structure, and X is just actin. And a similar question was on the Untitled paper. 39 wood sequence shows the routes taken by nitrogenous waste and insect from their production in the body cells to their removal from the insect's body. So this is a very obscure insect nitrogenous waste removal problem, and that's very annoying. The hemolymph is like blood. I feel like A is correct because the Malpighian tubules will absorb nitrogenous waste from the insect bloodstream, which is actually this hemolymph, into the entire gut, which processes both digesting food and also other wastes. They don't have kidneys. They don't have a nephron. The other choice is just ridiculously wrong, so I think A is a very clear choice here. Okay, last question: what function is maintained as a result of release of HCG? Well, HCG is released during early pregnancy so that the corpus luteum can be maintained. And the corpus luteum is a structure inside the ovary that produces progesterone, which maintains the uterine linings. Of these, a clear-cut correct answer. And so contrary to what I said during the video, I think this video will otherwise be too long if I go back and think of question 26. So let's just check the answer and I can accept my fate. Okay, moment of truth. First five's all good. D, A, D, C, D, so 10, I got it. I got this one wrong for some reason. Oh my God, that's not good. And 16, this is okay, but then I pretty much brought up the reason during the question. A, B, A, B. Wait, this is correct. And then 21 to 25 is all good. Oh, I got both correct. Okay, interesting. Last three, and then the rest correct. Okay, so I made my average worse. Again, another 30 out of 40. Again, excellent if you're a student, not so when you're a teacher. But let's check my question 15. I got this wrong. How did I get it wrong? They say it's D. Did I misread the number or something? 0.4%. What percentage of the energy passed from the primary producers to primary consumers? Yeah, so 40,000 is what's passed from primary producers to primary consumers. And then lost to cellular respiration by tertiary consumers. You lose 80. So 40 get passed on. The 40K get passed on, and then you lose 80. Do we have to go crunch the numbers in between as well? Did I misread something? Or should I be using the... Yeah, I think I just totally misread. So the 40K here does not account for this 20K that has been lost. So only 20K has been passed from the primary producers to primary consumers, and then 80 units of which being lost to respiration. So the mass should be 80 divided by 20K. Oh well, that's it. So it's not terrible while I figure out some of these very tough questions. But yeah, so oh well, it is what it is. I'll try to bounce back with the physics. I don't think many of you do biology and physics anyways, but yeah. But then for me, I'm a teacher. I need to be good at everything. So all right, see you in another video. Thanks for watching.